So welcome to the Department of Laboratory Medicine and Pathology Grand Rounds. It is my pleasure to introduce our speaker for today, Dr. Halling. Dr. Halling is a molecular pathologist and a professor in the Division of Laboratory Genetics. He is the vice chair of the oncology practice in the Genomics Laboratory. He received his MD and Ph.D. degrees from the University of Kansas and completed an anatomic and clinical pathology residency and clinical molecular genetics fellowship here at the Mayo Clinic. His primary area of interest is the development of molecular diagnostic tests that can be used for the diagnosis and the treatment of cancer patients. He and his team developed and implemented the Mayo Complete Solid Tumor Panel and are currently working on an RNA-seq assay that can be used to detect gene fusions in patients. Dr. Halling has published over 150 papers, mostly that relate to genetic testing of cancer. So thanks for being here today, Dr. Halling, and sharing your work. And now I will turn it over to you.
Thanks, Anne. Thanks for the nice introduction. And thanks for the opportunity to speak at the Grand Rounds. So as my talk says there, I'm going to talk about the evolution of molecular testing over the last several decades. And then we'll use that to move into a discussion of our Mayo Complete Solid Tumor Panel test. So I have no relevant financial disclosures for this presentation. All right, so these are the learning objectives for the talk. And so at the conclusion of this activity, you should be able to describe the evolution of clinical laboratory molecular testing over the past 40 years, discuss the types of alterations that are detected by solid tumor comprehensive genomic profiling, and then summarize the clinical utility of solid tumor comprehensive genomic profiling. All right. Here's an overview of my presentation. So I'm going to start by discussing the evolution of molecular testing, and in particular DNA sequencing over the last several decades. Then I'm going to show you how the evolution of DNA sequencing now allows us to comprehensively profile tumors for genetic alterations that can help in the management of cancer patients. And then we'll finish up by reviewing our findings for the first year of testing with our Mayo Complete Solid Tumor Panel. So first, let's talk a little bit about some things that we take for granted now. We haven't always known about DNA and RNA. They were actually first discovered in 1868 by a Swiss physician and biologist by the name of Friedrich Miescher. He isolated a new compound from the nuclei of white blood cells, and he showed that that was neither a protein, a lipid, or a carbohydrate, and therefore a novel biological molecule, which we now know are nucleic acids, which include both DNA and RNA. So he discovered DNA. But of course, he didn't know that that was the molecule that carried genetic information. It wasn't until the first half of the 20th century that various scientists got us to the point where we realized that DNA carried genetic material that would determine inheritance. And those included people like Frederick Griffith in the 1920s, Oswald Avery in the 1940s, and Alfred Hershey and Martha Chase in the 1950s. They're shown off to the right. Okay, the next really big thing, of course, was basically the discovery of the structure or the proposal for a structure of DNA by Watson and Crick in 1953. So they showed, of course, that DNA was composed of A's, C's, G's, and T's, deoxy, adenosine triphosphate, etc., and that they came together to form a double helical structure. But even despite that, way back in 1953, it was another 20 plus years before we could actually sequence DNA, easily sequence DNA. And that first occurred in 1976, when a couple scientists by the name of Maxim and Gilbert invented a sequencing method that would work for sequencing DNA. And I actually had the opportunity to try this out once in graduate school, and it was a very nasty technique. It used very noxious chemicals that you really don't want to use. And the results were hard to interpret. So I tried it once, and that was enough. Fortunately, a year later, a guy by the name of Frederick Sanger developed the technique that we now use a lot, and that's known as Sanger dideoxy sequencing. That was a big improvement over Maxim Gilbert sequencing and became the main type of DNA sequencing that was used until the 2010s. And we still use it today, but it's being somewhat supplanted by next-gen sequencing. This was such an important thing that Frederick Sanger received a Nobel Prize for this work in 1980, and it actually was his second Nobel Prize. The Sanger method for a long time utilized radioactive P32 or P33 labeled DATP, polyacrylamide gel electrophoresis, and autoradiography until about 2000. And then there was a switch to fluorescently labeled DNTPs and capillary electrophoresis in the late 1990s. And then along came next-gen sequencing in the mid-2000s, and that changed everything. But that was really not ready for prime time until the early 2010s when it started entering the clinical laboratories. So as I said, the ability to sequence DNA first became available about 1976, but it really was not an easy process either, all the way from about 1976 through 1983. It was slow, it was laborious, and all of this was because we didn't have PCR yet. And through 1985, and my graduate work involved sequencing, cloning and sequencing DNA. So it took me about 6 months to obtain about 2,000 bases of sequence for my PhD thesis in the early 1980s. And now you can easily get 2 billion bases of sequence in a day. So that would be about a million-fold increase. So why was it so difficult? Well, the reason is before PCR, we had to basically clone fragments of DNA to be able to create large enough quantities of pure clonal DNA to perform DNA sequencing. And that's what made it a long, laborious process. And this was the topic of my PhD thesis. Basically, what we did was, our goal was to isolate a gene known as ricin from the castor plant. It's a very toxic protein. And our goal was eventually to be able to clone it and express it and attach it to antibodies to cancer cells to perform an immunotoxin. That was a pretty ambitious goal, and we didn't get all the way there, but we were able to clone it and characterize it. And this shows the sequence that I generated for my thesis, and you can see it's just a little bit over 2,300 bases. It took a long time to get that. Then came PCR, and PCR was invented in 1983, and that changed everything. So PCR, or polymerase chain reaction, is basically a technique that allows you to create hundreds or billions of copies of a DNA fragment rather easily. It was one of the biggest advances in molecular biology that benefited both research and clinical genetic testing. And it was invented by Kerry Mullis in 1983 while he was working at the CETUS Corporation. So as I said, PCR allows one to easily create large quantities of a specific DNA fragment that can then be easily studied for mutations. And this avoided the need for gene cloning. It sped everything up. And Kerry Mullis for this invention received the Nobel Prize in 1993, and he talked about his work in this book here. All right, so this is what Sanger sequencing looked like for most of the 80s and most of the 90s. Basically, you needed a DNA template that was either created from cloning or PCR. You needed sequencing primer, you needed your deoxynucleotide triphosphates, you needed a radioactive DNTP, which, as I said, was usually P32 or P33 deoxy ATP. You needed a polymerase. And then we would create these large gels, and these gels were about the size of a computer monitor screen. So quite big, but they were very thin. They were about 4 millimeters thick. And they took a little practice to learn how to pour these gels and take them apart after the electrophoresis were done. So what we would do is we'd do our sequencing reactions and we load all the G's next to each other, the A's, the T's, and the C's, and electrophoresis in this direction. And then when it was done, we would separate the plates, put a filter paper on top of this and pull the gel off the plate. And then we would dry the gel. And then we would put it in an X-ray cassette with X-ray film to perform auto radiography. And the next day we'd come back and expose the film and then read our results. And the way it was read was we'd start at the bottom and we'd say, here's your G's. So there's 1, 2, 3 G's. So it'd be G, 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 A, T, C, T, T, etc. And you read all the way up. That's the DNA sequence. And as you can see, it's pretty easy to read at the bottom, but it gets hard to read at the top because everything gets scrunched together. So I did a lot of that when I was working with Dr. Thibodeau in the early 90s. So then it changed to fluorescent Sanger sequencing and capital electrophoresis in the later 1990s, and this made things a lot easier. So we no longer had to use radioactivity. Each of the nucleotides was labeled with a fluorescent, a fluorophore of a certain color. So T's were reds, C's were blue, etc. And you can see the distribution of things was even, unlike the gels where they get scrunched up at the top. So it made it easier to read a much longer. You could typically get 400 or 500 bases of sequence with this as compared to maybe 200 bases with the radioactive form. And the other thing is it sort of automates the calling of the sequence. It just auto tells you what you're reading here. And then you can compare a reference sequence to your patient sequence. And here you see right at this position, there's a change from a G to an A. So it's a G to A change. So this became the standard from about the late 1990s all the way to today. We still use this every day in the laboratory. With the improvements in sequencing, NIH decided that it would be a good idea to sequence the whole human genome, which is 3.2 billion bases. And that project was proposed to be a 15-year project. It started in 1990. And it was entirely done with traditional Sanger sequencing. There was no next-gen sequencing then. And it was completed in 2003. It actually became a race between NIH, which was led by Francis Collins, and Craig Venter, which was working at a company known as Solera. It was said to be about 99.9% accurate, but that sounds pretty accurate, but with 3.2 billion bases, that still leave quite a few errors. It only sequenced the euchromatic regions of the genome, which is about 90% of the human genome. Also, it was realized that Sanger sequencing was too slow, tedious, and expensive to rapidly sequence large genes, multiple genes, a whole exome or a whole genome, and that a new technology was needed both for patient care and research. And that's when NGS came along around in the mid-2000s. NGS, or next-gen sequencing, is also known as massive parallel sequencing or second-generation sequencing. And it basically allows us to do very high throughput DNA sequencing, enabling one to obtain billions of bases of sequence a day. So next-gen sequencing has been available since about 2005, but it's undergone constant improvement. And there's been a large number of companies that have been involved in this. Some of the early companies that developed this were Roche, and I think it was actually called Solexa way back when. I think Solexa got bought by Illumina. Then there was Life Technologies, which got bought by Thermo Fisher. And then there's some companies that have gone out of business. But they've all sort of driven this competition to make better and better next-gen sequencing. The predominant companies in this area now are Illumina and Thermo Fisher, and we mainly use Illumina platforms here. When I say platform, by the way, that refers to both the chemistry and the instrumentation. This graph sort of shows how quickly or how far we've come in the last 20 years with our ability to do DNA sequencing. So it's a growth of DNA sequencing. On the x-axis is the years, And over on the y-axis on the left, you can see this cumulative number of human genomes that have been sequenced over the years. And over on the right, it's the worldwide annual sequencing capacity in some very large numbers. So this would be terabases, which are as a trillion bases, petabases or quadrillion bases, exabases would be a quintillion, and this is sextillion. So what this is showing you is with the completion of the Human Genome Project, we had one genome that was sequenced. And it stayed that way for a little while until next-gen sequencing came along, and then there was sort of an exponential growth in our ability to sequence. And so by about 2012, we had 1,000 genomes sequenced. And back in 2017, that's 4 or 5 years ago now, we're up to about 1 billion, not 1 billion, but 1 million genomes that have been sequenced. And so if you extrapolate this out, the thought is that sometime during this decade, we should be able to sequence 1 billion genomes. Population of the world is about 7.7 billion, so that's a good fraction of the world population. With those improvements, sequencing became a lot less expensive. So it's estimated that the first human genome cost $100 million. And that cost has came down over the years, even faster than the drop in the cost of computer chips, which are described by Moore's law. So now it's estimated that you can sequence a genome for less than $1,000. So why am I telling you all this stuff about the evolution of DNA sequencing? Well, the reason is to lead you up to the next thing, which is to show you that the advances in DNA sequencing over the past 40 years now allow us to rapidly and relatively inexpensively obtain large amounts of DNA sequencing data from patients' tumors. And we're now calling, this has now been called comprehensive genomic profiling, even though that's a little bit of a misnomer. But we can do this thing called comprehensive genomic profiling of tumors for genetic alterations. And this is important because tumors tend to have a lot of genetic alterations, and many of these alterations have potential clinical utility for things such as making the right decisions about therapy, determining a patient's prognosis, or helping pathologists establish a definitive diagnosis for a tumor. Okay, just a little basic review. I usually show this slide in every talk. I think you all realize that cancer is a genetic disease and that the research over the past 50 years has revealed that cancer results from mutations in cancer-causing genes known as oncogenes and tumor suppressor genes. Out of the 20,000 or so protein encoding genes in the human genome, it's estimated that there's about 500 to 1,000 that have the potential to contribute to cancer when mutated. Now, having said that, not all 500 to 1,000 genes are going to be mutated in any given patient's tumor. Usually it's a much smaller number, maybe 5 or 10 that are critically important. But if you look across all cancers, there's been about 500 to 1,000 that have been shown to have ability to promote tumorigenesis. Again, the cancer genes are either referred to as oncogenes or tumor suppressor genes. And oncogenes promote tumorigenesis through activating mutations, and tumor suppressor genes promote tumorigenesis through inactivating mutations. Now, mutations come in many different forms, and I like to divide them into mutations that you can see at the chromosome level, or mutations you see at the molecular genetic level. And examples of chromosomal level mutations are chromosomal translocations, such as the ones shown off to the right, which is the Philadelphia chromosome that occurs in chronic myelogenous leukemia. That's the very first genetic abnormality that was associated with the cancer way back in the 60s. Other types of chromosomal level mutations that are very important is copy number variants. And these refer to either chromosomal gains or losses, either entire chromosomes or regions of chromosomes. And then sometimes we say just regions of amplification of genes, so genes that are highly amplified just locally. So for instance, HER2 amplification in breast cancer. So that's one class, chromosome 11 mutations, and those are typically picked up with tests such as chromosomal microarray or FISH or karyotyping. But now next-gen sequencing can do some of this too. And then there are molecular genetic level mutations, things we can't see at the chromosomal level. We refer to those as submicroscopic mutations. And the main types of molecular genetic mutations are single nucleotide variants, abbreviated as SNVs, and we used to refer to these as point mutations. And then there's small insertions and deletions, which we refer to as indels. Okay, so it wasn't until about a year ago that we were able to comprehensively profile tumors. So until recently, we've just been offering tests for mutations in individual genes, such as the KRAS gene or the EGFR gene, or offering small targeted NGS panels. Our very first NGS panel, which I think was introduced maybe about 2015, was a 50 gene. 2015 was a 50 gene panel. And we're still using that today. But in April 2021, we went live with what we refer to as a large cancer panel that we now call Mayo Complete Solid Tumor Panel, or MCSDP. And that allows us to do comprehensive genomic profiling. So when we say comprehensive genomic profiling, what do we mean? Well, it's not totally comprehensive. So comprehensive, according to what Merriam-Webster means, including many, most, or all things. And when we see CGP, we mean a very large NGS panel that assesses for genetic alterations, for example, single nucleotide variants, indels, copy number variants, gene fusions, and splice variants in a very large number of genes, generally over 300 genes. And if you look at other laboratories' offerings for CGP, they all have probably in the 300 to 600 range, 300 to 600 gene range represented on their panels. Okay, so what's not assessed by our CGP tests? Well, they're not whole genome sequencing, and so much of the genome is not assessed. In fact, we're only sequencing about 2 megabases out of 3.4 gigabases. So 2 million bases out of about 3.4 billion. So there's a lot of DNA that's not assessed. And we're not looking for other sorts of alterations, such as methylation or gene expression. That requires a different type of test. You may ask the question, well, why aren't we doing whole exome or whole genome sequencing? Well, you get a lot of interesting information in that, but it's very expensive. And it's sort of hard or impossible to do when you have very small quantities of tissue. And a lot of our specimens that we get from our clinicians and patients are very small biopsies. So there's a limit to how big you want to go if you want to be able to handle most specimens. We also, when you do whole exome or whole genome, you get a lot of information that we don't know how to interpret yet due to uncertain clinical significance. So we think, 300 to 600 genes is sort of the sweet spot for large panels. All right, so this is our test, which we call Mayo Complete Solid Tumor Panel, or MCSDP. And this shows what it assesses for off to the right. So it utilizes Illumina's TSO 500 chemistry and pipeline. But, we couldn't just take it out of the box and start using it. We had to do a lot of work, a year and a half of work to validate the test, to establish performance characteristics such as accuracy, reproducibility, analytical sensitivity, all that stuff. It really is it's 6 tests in one, and it utilizes both DNA and RNA. And so for the DNA part of the test, what we're able to do is look for SNVs and indels in 514 genes. We're able to look at gene amplification. It's the same thing as copy number alteration in 59 genes. Very importantly, and I'll talk about this in several slides coming up, we're able to assess the tumor for its tumor mutational burden, basically how highly mutated is the tumor. We're also able to determine if the tumor exhibits microsatellite instability. So we get all that from the DNA side of the test. And then on the RNA side, we're able to look for gene fusions in 55 genes and splice variants in 3 genes. This test is validated on paraffin-embedded specimens and also cytology sites. And our validation of cytology sites is a nice thing because most of our competitors don't offer that. As I've already mentioned, the clinical utility of the test is primarily for helping oncologists make decisions about therapy. But secondarily, we oftentimes get information that's helpful for establishing a diagnosis or predicting a patient's prognosis. Before we even started developing or moving forward with validation of this test, we were a little worried about the number of mutations we'd find in tumors and how we would keep up with the reporting, because there's oftentimes a lot of research that goes into figuring out what's the clinical significance of a mutation. So we spent a lot of time evaluating what we call genomic interpretation tools to see if they could help us. And there were probably 11 or 12 companies who had software packages that we looked at. And we eventually settled on one from QIAGEN called QIAGEN Clinical Insights that helps us with variant interpretation and clinical trial information. A few final notes on this slide. So again, I want to reiterate that one of the greatest values of this test is its ability to determine TMB, or tumor mutational burden. You cannot do that with small panels. You generally need a test that has at least 300 genes to accurately determine TMB. The other point I want to make, and it would help you with one of the pre-test questions, is that this test is not designed to determine the primary site for metastatic tumors of unknown origin. So oncologists sometimes have patients where they have a patient with metastatic tumor and they don't know where the tumor originated from. Was it the prostate, the breast, the lung, or wherever? And there are tests that can help you sort of determine that, but this test wasn't really designed for that. Nonetheless, we occasionally do get information that gives us clues as to where the tumor is originating from. These are the 514 genes on the panel. And the way this thing was designed was it designed to provide 100% coverage for the National Comprehensive Care Network guidelines. In other words, this group, the National Comprehensive Cancer Network, provides recommendations on genes that need to be tested for various tumor types. And so we have NCN coverage for 11 tumor types. And then we also have coverage of genes involved in 1,600 clinical trials. Okay, as I said a couple slides ago, we knew that the big job is, once we had the test line, was going to be able to interpret the mutations. The term we use these days are variants or genetic alterations. So how are we going to interpret these? And basically, we follow guidelines that were established by AMP, ASCO, and CAP way back in 2017. And what they recommended in their white paper was that variants be categorized as either tier 1, 2, 3, or 4. Tier 1 variants are variants that have strong clinical significance, either therapeutically, diagnostically, or prognostically. Tier 2 variants are those that have potential clinical significance. Tier 3 are variants that have unknown clinical significance. And Tier 4 are benign variants. So in our reports, we report all the tier 1 through 3 variants, but we only provide, so we report them, but we only interpret the tier 1 and 2 variants. And we do not include the tier 4 variants. Typically, on any given patient's tumor, we see about 3 to 7 tier 1 or 2 variants, as long as the case is not hypermutated. If it's hypermutated, we can see a lot more than 3 to 7. This is a typical report. And so we provide what we call a result table. This patient had lung adenocarcinoma. In the first column, we list the variants that we saw. Second column, we list how those variants are categorized by tier. In the third column, we say whether there are any FDA-approved therapies for that variant. We never We no longer provide this column, so I'm not going to talk about it. And then the last thing we provide is information on whether there are clinical trials. So if there isn't an FDA-approved therapy, such as this alteration, BAP1, there's no FDA-approved therapy there, but there is a clinical trial. So a patient could be still entered into a trial that would investigate a new drug for that alteration. So for this patient, they had a high TMB, and there is an FDA-approved therapy that's a tier 1A variant, which has an FDA-approved therapy. They also had an EGFR mutation, for which there is also an FDA-approved therapy. And as I said, so if there are clinical trials available for a variant, we'll list those at the end of the report. So we'll list the variant, and then we'll list the trial ID, the title of the trial, and any contact information. And oncologists like this a great deal, especially at academic institutions like Mayo, where they're constantly trying to enter patients into clinical trials. Coming back to the description of the test, so what is the clinical utility of each of these alterations? Well, I want to focus on the clinical utility of tumor mutational burden first. Again, a reminder that TMB cannot be determined with small panels, and you need a big panel like this. So in our tests menu, this is the only test that can determine TMB. TMB, or tumor mutational burden, is categorized as TMB high or high levels of tumor mutations if there are more than 10 mutations per megabase of sequenced DNA. And it's TMB low if it's less than 10 mutations per megabase. The reason it's important to determine the TMB status of a tumor is there's a lot of drugs that have been developed in the last 5 to 10 years, which we refer to as immunotherapies, that tend to work well on patients that have high tumor mutation burden. Final note I want to say is our test, of course, also assesses for microsatellite stability. And if there are high levels of microsatellite stability, the tumor will, but the reverse is not true. A tumor can show a high TMB, but not microsatellite stability. All right, so it's important to realize that tumors vary in the number of mutations that they carry or their tumor mutational burden. This is a famous plot from early 2010s that assessed a bunch of different tumor types for their tumor mutational burden using whole exome sequencing. So on the x-axis, you see various tumor types. And on the y-axis is the somatic number of mutations per megabase. And this is exponential here. This is 0.0. This would be 1 mutation per megabase, 10 mutations per megabase, 100, et cetera. And each of these dots is a different patient's tumor. And so the red lines represent the median number of mutations for that tumor type. And so you can see melanomas tend to be the most highly mutated tumors, along with lung cancers. Tumors that have very few mutations include things like CLL, thyroid tumors, kidney tumors, et cetera. This difference may not look as striking as it is, but remember, this is an exponential scale. So again, to remind you, tumors with high TMBs have been shown to have better responses to immunotherapy, such as Opdivo and Keytruda. And the reason is presumably because they have more neoantigens, which allow a patient's immune system to attack the tumor. And that's a topic in and of itself, which would take another whole talk to talk about. So I can't go too deeply into it. This was an interesting study, and this gets back to the relationship between microsatellite stability and tumor mutational burden. In this study, they actually looked at a huge number of tumors, 62,000 tumors, and they found that 4,300 of them had high tumor mutation burden and 699 showed microsatellite stability. And as I said, almost all tumors that show microsatellite stability will also have a high TMB, but the reverse is not true. And the reverse is particularly not true in tumors like lung cancer and melanoma. Usually when you see high TMB, it's not due to microsatellite stability, but due to things like carcinogens and tobacco smoke or sunlight, UV sunlight. All right, so there's been a lot of important FDA approvals of drugs over the last 5 years, which require testing a tumor for the types of alterations that we detect with our large cancer panel, our MCSTP assay. So in 2017, the FDA gave FDA approval for pembrolizumab, which is an immunotherapy, for advanced solid tumors, regardless of where the tumor is coming from. So the histology really doesn't make any difference. Could be a breast cancer, could be lung cancer. As long as the tumor showed microsatellite stability, it was found that it responded well to pembrolizumab. So that's why testing our tumors for microsatellite stability is important. A year later, it was FDA approved a drug called larotrectinib for any solid tumor that had an NTRK gene fusion. And that's another thing that we test for on our panel. And then in 2020, FDA approved pembrolizumab for any tumor, advanced tumor in adults and adults and children that had a high TMB. And of course, as I said, that's another thing we test for. So our large cancer panel will cover us from all these things: microsatellite stability, NTRK fusions, and tumor mutation burden. All right, so we talked a lot about tumor mutation burden, but is there any utility for all those 514 genes looking for SNVs and indels? And the answer is yes. There's a lot of targeted therapies that have been developed over the years that are now FDA approved that are useful if patients have certain mutations. So one of the first ones was mutations in EGFR in lung cancer were shown to respond to certain drugs, such as erlotinib and afatinib. More recently, it's been shown that patients with lung cancer that have KRAS G12C mutations respond to a drug. This is actually incorrect. It should be sotorasib. And the list goes on. So there are a lot of FDA-approved therapies for mutations, SNVs and indels, and various oncogenes. All right, now I'm going to talk a little bit about our experience with the test over the last year. We've actually done a little bit over 500 cases, but we have data here for about 427. And of the first 427 patients that we did the testing for, about 70% of them were Mayo patients and about 30% were MCL patients. And here shows the age distribution for the patients that we've sequenced. And you can see most of these patients are older, which is expected for cancer patients. You know, it looks like it maxes out in the 60s and 70s. We were interested in how often we were seeing patients who had a high tumor mutation burden or microsatellite stability. And I think I mentioned that our cutoff for TMB high is 10 mutations per megabase or higher. And so you can see that about 70% of the patients were TMB low, and about 30% were TMB high. Probably the patients that are most likely to respond to immunotherapy would be this group, these ones with 20 mutations per megabase or more, and about 9% of patients fell into that group. Interestingly, only about 1% of our patients show microsatellite stability. And I think that's due to the fact that so many of our patients were lung cancer patients, and microsatellite instability is rare in that tumor type. Microsatellite stability is pretty common in colon cancers and endometrial cancers and gastric cancers, but those are a small proportion of all our testing. And so I think the low frequency of microsatellite instability is due to the fact that we are mostly analyzing lung cancers. This slide actually shows the types of tumors that we did analyze. So if you look at all the patients, all 427, you can see that about 60% of them were lung cancer patients. But we did do other tumor types, such as some sarcomas, pancreatic cancer, melanoma, etc. If we separated this out in just our Mayo patients versus our MCL patients, we saw something interesting, and that is 80% of our patients from Mayo are lung cancer patients, Whereas for our MCL clients, there's a much broader mix of tumor types that we analyzed. We are also interested in knowing how many tier 1 and 2 variants we were seeing per case. And it maxed out at about 2. So about 23% of our patients showed 2 variants. About 18% showed 3 variants. But anyway, you can see that most patients show somewhere between 1 and 6 variants per case. And that's a pretty manageable number in terms of trying to create a report for it. Interestingly, we had 4% of our patients, or actually 19 patients with about 4% with no alteration. With such a large panel, we always get a little worried when we don't see anything. So we always go back to the H&E slide and make sure the tumor percent was adequate. We had enough tumor. And but tumors can occasionally show no alterations with our test. And so we just reassured ourselves by double checking everything. Relatively few patients had 10 or more variants, was only about 1%. And those are the ones that are a little bit more challenging to create a report for. It can be quite a long report. These are generally patients that had high tumor mutation burden. And sometimes with the high TMB cases, we'll just provide interpretations for the tier 1 variants, because that usually gives the clinician more than enough to work with. Okay, if you look, and if you ask the question, what fraction of the patients that we analyzed was there an FDA-approved targetable variant for? It was, so there were 427 patients, 182 had a variant for which there was an FDA-approved therapy. And that leaves about 57% that didn't have a targetable variant, FDA-approved targetable variant. But many of these patients could be entered into clinical trials. If we look at the variants that were targetable, a high proportion of them were cases that showed a high tumor mutation burden. But there were a lot of SNVs and indels that were also targetable too, such as mutations in EGFR, MET exon skipping mutations, PIK3CA mutations in breast cancer, etc. So if we took out the TMB and MSIH cases, you can see that 23% of the patients would have had a targetable therapy, FDA-approved therapy. All right, I'm going to finish up my talk now with a few case studies that demonstrate the clinical utility of the test for specific patients. So this first patient was a 79-year-old female. She had cerebral adenocarcinoma. That's a tumor of the proximal colon. Her tumor demonstrated a very high tumor mutation burden with 54.6 mutations per megabase. It also exhibited microsatellite instability. So basically, the reason this patient is showing a high tumor mutation burden is because they have microsatellite instability, which results from defective DNA mismatch repair. So this clinical significance of that is that this patient is a good candidate for immunotherapy. Patient also showed a BRAF V600E mutation. So BRAF is an oncogene. It's a very common mutation in that oncogene. And that actually, one of the importance of this is that when we see a microsatellite instability phenotype in a colon cancer, you have to think about the possibility the patient may have hereditary colon cancer syndrome known as Lynch syndrome. But if you see this mutation, that makes Lynch syndrome unlikely. So that's helpful information. We also saw KRAS and NRAS mutations, and those would predict that the patient would not be likely to respond to anti-EGFR therapies. So a lot of good information came from sequencing this patient's tumor with the MCSTP assay. The second case is an example of where we got some interesting copy number alteration data. So this was a 78-year-old male that has esophageal adenocarcinoma. It had 11.8 mutations per megabase, which would tip it over into the TMB high category, but barely. I wouldn't predict that immunotherapy would be really effective here, but you never know. It was also a microsatellite stable tumor. But the interesting thing was when we look at the copy number information, which is shown down below here, we saw amplification of a region on the long arm of chromosome 11, which contained these 4 genes, cyclin D1, fibroblast growth factor 19, et cetera. It's FGF19, FGF3, FGF4. And so the way you read these plots here is the different chromosomes are listed from left to right. So you got chromosome 1, 2, 3, 4, 5. And each of these dots represent a different gene on the panel. So for our 514 genes, if there is no change in the copy number, if it's a normal copy number, it will hover around this 0 line here. So if you look across here, you can see there is loss of the short arm of chromosome 1. There's gain of chromosome 13 at 20 here. But most interestingly is this region of amplification of several genes. And these are these 4 genes mentioned right here. There is not an FDA-approved therapy for amplification of any of these genes, but there are a couple clinical trials for tumors that show CCND1 amplification, and we mentioned those in the patient's report. So this patient has potential to be entered into those clinical trials. Here's a case that demonstrates the utility of the test for picking up gene fusions. This was a 69-year-old female with metastatic lung cancer, adenocarcinoma. Her tumor showed a low-level TMB, only 4.7 mutations per megabase, but it did show an important fusion. So there was a fusion, gene fusion between 2 genes known as KIF5B and the red oncogene. This is a tyrosine kinase. And this is very important to find because there's an FDA-approved therapy that targets this fusion. Specifically, it targets the red part of this fusion. And the FDA-approved therapies are known as selpercatinib and pralsetinib, and they received FDA approval a few years ago. So that was very important for this patient. Interestingly enough, this patient, as I said, did have a low tumor mutation burden. And it's been found that there usually when you see these fusions and other fusions we sometimes see in lung cancer, ALK fusions and red ROS fusions. So ALK, ROS, and red are common fusions. And there's FDA-approved therapies for all of those. But almost always those tumors show low tumor burden, so are not candidates for immunotherapy. Okay, the last case is just an example of how occasionally this test will provide information that's helpful for establishing a definitive diagnosis. And this was an 11-year-old boy who had a type of sarcoma that was described as being consistent, histologically consistent with spindle cell or sclerosing rhabdomyosarcoma. But they wanted to know whether the tumor had a mutation, a specific mutation in a gene known as Mayo D1. The mutation was a leucine to an arginine chain change at codon 122. This mutation in this gene is specific for that tumor type. And so they ordered that there was no testing for this anywhere across the country as a single gene test. So they ordered our test, even though they only wanted this one gene. And then we did find that mutation, so it helped definitively establish that diagnosis. All right, so that's my last slide, next to last slide, but I just want to thank all the people that helped develop and implement this test. It took a lot of lab directors and pathologists, development technologists. A special thanks to Beth Patel, who helped create some of the slides for me today. A lot of great work by our development tech, Shannon Knight, Jesse Voss, Falcona Milosevic, Joe Thompson, Joe Blamo, Calvin Jurdy, and Eric Supperman. And basically, these were development techs in the genomics lab, but also CGSL and ADL. So very collaborative effort. The bioinformaticians were critical, and we started this project with Surendra, did a lot of great work. It still does. And then it was transferred over to Gopi, Jag, and Kim, who've done most of the clinical work with the test. Great work from our technical specialists. Genetic counselors are so key for this test. They they really do most of the work when it comes to creating reports and interpreting results. A lot of thanks to Jessica, Amber, Lisa, Lindsay, Antonina, and Claire. I think all of our molecular oncology GCs are now trained on putting reports together for this test. And of course, we couldn't implement this test without our management group, critical group, and then the IT group. And I probably have not completely listed all the IT group that was involved here, but special thanks to Tony Stye and Nate Matson. And then Alyssa is our oncology product manager for NCL, and she's done a wonderful job with the test too. So to conclude, I think you can see that DNA sequencing has come a long way since its advent way back in 1976. And it's become particularly powerful in the last 10 years. So it's much more powerful than it was even 10 years ago. And the availability of next-gen sequencing allows us to comprehensively profile tumors for genetic alterations that can predict response to therapy, establish a diagnosis, and predict prognosis. And CGPs are particularly important for determining tumor mutational burden, which helps predict whether patients will respond to immunotherapies. But the identification of SNVs and indels and copy number alterations is also very important. And finally, I would say that even when we don't find an FDA-approved a variant that has an FDA-approved therapy, we can still find variants that allow us to enter patients into clinical trials and validate new drugs. So,
Thank you for participating. Please click the button below to complete the evaluation and obtain credit.